This video will discuss the fundamental process of recombinant DNA cloning. Observe the steps shown in this image. Although this is a complex process, this video will break down the steps to a more simplistic understanding using very simple designs made of simple Play-Doh. First, observe the time lapse of our model recombinant DNA process. We first start off with a double-stranded piece of DNA. This piece of DNA would have many genes on it, each with a different purpose. Suppose that you want to make a copy of only a specific gene on this piece of DNA. This gene is pictured here in blue and will be our primary focus in this video. Once you have determined which gene you want to copy, you must find a way to remove this gene from the DNA strand. Restriction enzymes are specially designed enzymes that scan over the DNA strand looking for a specific sequence. Once it locates the correct place, it attaches to one end of the DNA strand. Another restriction enzyme will then attach at the opposite end. These enzymes act like a pair of scissors to cut out the specific gene that you're looking for. Now, the gene has been cut by restriction enzymes and is isolated by itself. At this point, it is ready to move to the next step of the process, genetic recombination. The specific gene will be pasted into a plasmid. The plasmid is a genetic structure that is useful in gene manipulation because it can replicate independently of the chromosomes in a cell. In order to select for the correct gene, the plasmid will also contain an antibiotic gene which assists in identifying the specific gene you are attempting to replicate. The gene will then be inserted onto the plasmid by matching up the ends of the gene with the correct base pairs in the plasmid. The enzyme DNA ligase acts as a glue to keep these base pairs together by creating a phosphodiester bond between the two molecules. Once bonding has occurred between the gene and plasmid, the plasmid is introduced into a solution with bacteria, such as E. coli. A heat shock is applied to the solution, which causes the bacteria to take in some of the plasmids. This allows the gene to be cloned by taking advantage of the bacteria replication. After the heat stroke has been applied, the solution is added to an agar plate that has been treated with nutrients and antibiotics. The nutrients allow the bacteria to grow in colonies and replicate. Because the agar plate has also been treated with antibiotics, bacteria colonies without the antibiotic-resistant gene will die, leaving only the bacteria containing the plasmid and gene that is desired to continue to grow and replicate.